Hey students, this is your Nandini Ma'am. Today we are going to read chapter number 5, Molecular Basis of Inheritance of Class +2, Board CBSE, and the book is NCERT. So let's get started. In the previous chapter, you have learned the inheritance patterns and the genetic basis of such patterns. At the time of Mendel, the nature of those factors regulating the pattern of inheritance was not clear. Over the next 100 years, the nature of the putative genetic material was investigated, culminating in the realization that DNA, deoxyribonucleic acid, is the genetic material, at least for the majority of organisms. In class 11th, you have learned that nucleic acids are polymers of nucleotides. Deoxyribonucleic acid, DNA, and ribonucleic acid, RNA, are the two types of nucleic acids found in living systems. DNA acts as the genetic material in most of the organisms. RNA, though it also acts as a genetic material in some viruses, mostly functions as a messenger. RNA has additional roles as well. It functions as adapter, structural, and in some cases as a catalytic molecule. In class 11, you have already learned the structures of nucleotides and the way these monomer units are linked to form nucleic acid polymers. In this chapter, we are going to discuss the structure of DNA, its reapplication, the process of making RNA from DNA, transcription, the genetic code that determines the sequences of amino acids in proteins, the process of protein synthesis, translation, and elementary basis of their regulation, the determination of complete nucleotide sequence of human genome during last decade has set in a new era of genomics. In the last section, the essentials of human genome sequencing and its consequences will also be discussed. Let us begin our discussion by first understanding the structure of the most interesting molecule in the living system, that is, the DNA. In subsequent sections, we will understand that why it is the most abundant genetic material and what its relationship is with RNA. 5.1. The DNA. DNA is a long polymer of deoxyribonucleotides. The length of DNA is usually defined as number of nucleotides or a pair of nucleotide referred to as base pairs present in it. This also is the characteristic of an organism. For example, a bacteriophage known as phi into 174 has 5,386 nucleotides. Bacteriophage lambda has 48,502 base pairs. Escherichia coli has 4.6 into 10 to the power 6 base pairs, and haploid content of human DNA is 3.3 into 10 to the power 9 base pairs. Let us discuss the structure of such a long polymer. 5.1.1 Structure of polynucleotide chain. Let us recapitulate the chemical structure of a polynucleotide chain, DNA or RNA. A nucleotide has three components: a nitrogenous base, a pentose sugar, ribose in case of RNA, and deoxyribose for DNA, and a phosphate group. There are two types of nitrogenous bases, purines, adenine and guanine, and pyrimidines, cytosine, uracil, and thymine. Cytosine is common for both DNA and RNA, and thymine is present in DNA. Uracil is present in RNA at the place of thymine. A nitrogenous base is linked to the OH of 1' carbon pentose sugar through a nitrogen glycosidic linkage to form a nucleoside, such as adenosine or deoxyadenosine, guanosine or deoxyguanosine, cytidine or deoxycytidine, and uridine or deoxythymidine. When a phosphate group is attached or linked to OH of 5' carbon of a nucleoside through phosphoester linkage, a corresponding nucleotide or deoxynucleotide, depending upon the type of sugar present, is formed. Two nucleotides are linked through 3' 5' phosphodiester linkage to form a dinucleotide. More nucleotides can be joined in such a manner to form a polynucleotide chain. A polymer has formed at one end a free phosphate moiety at 5' end of sugar, which is referred to as 5' end of polynucleotide chain. Similarly, at the other end of the polymer, the sugar has a free OH of 3' carbon group, which is referred to as 3' end of the polynucleotide chain. The backbone of a polynucleotide chain is formed due to sugar and phosphates. The nitrogenous bases linked to sugar moiety project from the backbone. Figure 5.1. In RNA, every nucleotide residue has an additional OH group present at 2' position in the ribose. Also, in RNA, the uracil is found at the place of thymine, 5-methyl uracil, another chemical name for thymine. DNA as an acidic substance present in nucleus was first identified by Friedrich Miescher in 1869. He named it as nuclein. However, due to technical limitations in isolating such a long polymer intact, the elucidation of structure of DNA remained elusive for a very long period of time. It was only in 1953 that James Watson and Francis Crick, based on the X-ray diffraction data produced by Maurice Wilkins and Rosalind Franklin, proposed a very simple but famous double helix model for the structure of DNA. One of the hallmarks of their proposition was base pairing between the two strands of polynucleotide chains. However, this proposition was also based on the observation of Erwin Chargaff that for a double-stranded DNA, the ratios between adenine and thymine and guanine and cytosine are constant and equals 1. The base pairing confers a very unique property to the polynucleotide chains. They are said to be complementary to each other, and therefore, if the sequence of bases in one strand is known, then the sequence in other strand can be predicted. Also, if each strand from a DNA, let us call it as a parental DNA, acts as a template for synthesis of a new strand, the two double-stranded DNA, let us call them as daughter DNA, thus produced would be identical to the parental DNA molecule. Because of this, the genetic implications of the structure of DNA became very clear. The salient features of the double helix structure of DNA are as follows. Number 1. It is made up of two polynucleotide chains where the backbone is constituted by sugar phosphate and the bases project inside. Number 2. The two chains have anti-parallel polarity. It means if one chain has the polarity 5' to 3', the other has 3' to 5'. Number 3. The bases in two strands are paired through hydrogen bond, forming base pairs. Adenine forms two hydrogen bonds with thymine from opposite strand and vice versa. Similarly, guanine is bonded with cytosine with 3 hydrogen bonds. As a result, always a purine comes opposite to a pyrimidine. This generates approximately uniform distance between the two strands of the helix. Figure 5.2. Number 4. The two chains are coiled in a right-handed fashion. The pitch of the helix is 3.4 nm. A nanometer is 1 billionth of a meter, that is 10 to the power minus 9 meters, and there are roughly 10 base pairs in each turn. Consequently, the distance between a base pair and a helix is approximately 0.34 nanometer. Number 5. The plane of one base pair stacks over the other in double helix. This, in addition to hydrogen bonds, confers stability of the helical structure. Figure 5.3. Compare the structure of purines and pyrimidines. Can you find out why the distance between the two polynucleotide chains in DNA remains almost constant? The proposition of a double helix structure for DNA and its simplicity in explaining the genetic implication became revolutionary. Very soon, Francis Crick proposed the central dogma in molecular biology, which states that the genetic information flows from DNA towards RNA towards protein. In some viruses, the flow of information is in reverse direction, that is, from RNA to DNA. Can you suggest a simple name to the process? 5.1.2 Packaging of DNA helix. Taken the distance between two consecutive base pairs as 0.34 nanometer, if the length of DNA double helix in a typical mammalian cell is calculated simply by multiplying the total number of base pairs with distance between two consecutive base pair, that is 6.6 into 10 to the power 9 base pair multiplied by 0.34 into 10 to the power minus 9 meter per base pair. So it comes out to be approximately 2.2 meters, a length that is far greater than the dimension of a typical nucleus, approximately 10 to the power minus 6 meter. How is such a long polymer packaged in a cell? If the length of E. coli DNA is 1.36 millimeter, can you calculate the number of base pairs in E. coli? In prokaryotes such as E. coli, though they do not have a well-defined nucleus, the DNA is not scattered throughout the cell. DNA, being negatively charged, is held with some proteins that have positive charges in a region termed as nucleotide. The DNA in nucleoid is organized in large loops held by proteins. In eukaryotes, this organization is much more complex. There is a set of positively charged basic proteins called histones. A histone acquires charge depending upon the abundance of amino acids residues with charged side chains. Histones are rich in the basic amino acid residues, lysine and arginine. Both the amino acid residues carry positive charges in their side chains. Histones are organized to form a unit of 8 molecules called histone octamer. The negatively charged DNA is wrapped around the positively charged histone octamer to form a structure called nucleosome. Figure 5.4a. A typical nucleosome contains 200 base pairs of DNA helix. Nucleosomes constitute the repeating unit of a structure in nucleus called chromatin, thread-like stained bodies seen in nucleus. The nucleosomes in chromatin are seen as beads on string structure when viewed under electron microscope. Figure 5.4b. Theoretically, how many such beads, nucleosomes, do you imagine are present in a mammalian cell? The beads on string structure in chromatin is packaged to form chromatin fibers, which are further coiled and condensed at metaphase stage of cell division to form chromosomes. The packaging of chromatin at higher level requires additional set of proteins that collectively are referred to as non-histone chromosomal proteins. In a typical nucleus, some region of chromatin are loosely packed and stains light and are referred to as euchromatin. The chromatin that is more densely packed and stains dark are called as heterochromatin. Euchromatin is said to be transcriptively active chromatin, whereas heterochromatin is inactive. 5.2. The search for genetic material. Even though the discovery of nuclein by Miescher and the proposition for principles of inheritance by Mendel were almost at the same time, but that the DNA acts as a genetic material took long to be discovered and proven. By 1926, the quest to determine the mechanism for genetic inheritance had reached the molecular level. Previous discoveries by George Mendel, Walter Sutton, Thomas Hunt Morgan, and numerous other scientists had narrowed the search to the chromosomes located in the nucleus of most cells. But the question of what molecule was actually the genetic material had not been answered. Transforming principle. In 1928, Frederick Griff, in a series of experiments with Streptococcus pneumoniae, bacterium responsible for pneumonia, witnessed a miraculous transformation in the bacteria. During the course of his experiment, a living organism had changed in physical form. When Streptococcus pneumoniae bacteria are grown on a culture plate. Some produce smooth, shiny colonies while others produce rough colonies. This is because the S strain bacteria have a mucus coat while R strain does not. Mice infected with the S strain virulent die from pneumonia infection, but mice infected with the R strain do not develop pneumonia. Griffith was able to kill bacteria by heating them. He observed that heat killed S strain bacteria injected into mice did not kill them. When he injected a mixture of heat killed S and live R bacteria, the mice died. Moreover, he recovered living S bacteria from the dead mice. He concluded that the R strain bacteria had somehow been transformed by the heat killed S strain bacteria. Some transforming principle transferred from the heat killed S strain had enabled the R strain to synthesize a smooth polysaccharide coat and become virulent. This must be due to the transfer of the genetic material. However, the biochemical nature of genetic material was not defined from his experiments. Biochemical characterization of transforming principle. Prior to the work of Oswald Avery, Colin McLeod, McLean McCarthy, the genetic material was thought to be a protein. They worked to determine the biochemical nature of transforming principle in Griffith's experiment. They purified biochemicals from the heat-killed S cells to see which ones could transform live R cells into S cells. They discovered that DNA alone from S bacteria caused R bacteria to become transformed. They also discovered that protein digesting enzymes and RNA digesting enzymes did not affect transformation. So, the transforming substance was not a protein or DNA or RNA. Digestion with DNAs did inhibit transformation, suggesting that the DNA caused the transformation. They concluded that DNA is the hereditary material, but not all biologists were convinced. Can you think of any difference between DNAs and DNAs? 5.2.1 The genetic material is DNA. The unequivocal proof that DNA is the genetic material came from the experiments of Alfred Hershey and Martha Chase. They worked with viruses that infect bacteria called bacteriophages. The bacteriophage attaches to the bacteria and its genetic material then enters the bacterial cell. The bacterial cell treats the viral genetic material as if it was its own and subsequently manufactures more virus particles. Hershey and Chase worked to discover whether it was protein or DNA from the viruses that entered the bacteria. They grew some viruses on a medium that contained radioactive phosphorus and some others on medium that contained radioactive sulfur. Viruses grown in the presence of radioactive phosphorus contained radioactive DNA but not radioactive protein because DNA contains phosphorus but protein does not. Similarly, viruses grown on radioactive sulfur contain radioactive protein but not radioactive DNA because DNA does not contain sulfur. Radioactive phages were allowed to attach to E. coli bacteria. Then, as the infection proceeded, the viral coats were removed from the bacteria by agitating them in a blender. The virus particles were separated from the bacteria by spinning them in a centrifuge. Bacteria, which was infected with viruses that had radioactive DNA, were radioactive, indicating that DNA was the material that passed from the virus to the bacteria. Bacteria that were infected with viruses that had radioactive proteins were not radioactive. This indicates that proteins did not enter the bacteria from the viruses. DNA is therefore the genetic material that is passed from from virus to bacteria. Figure 5.5. 5.2.2 Properties of genetic material DNA versus RNA. From the foregoing discussion, it is clear that the debate between proteins versus DNA as the genetic material was unequivocally resolved from Hershey-Chase experiment. It became an established fact that it is DNA that acts as a genetic material. However, it subsequently became clear that in some viruses, RNA is the genetic material. Answer to some of the questions, such as why DNA is the predominant genetic material, whereas RNA performs dynamic functions of messenger and adapter, has to be found from the differences between chemical structures of the two nucleic acid molecules. Can you recall the two chemical differences between DNA and RNA? A molecule can act as a genetic material, must fulfill the following criteria. Number one, it should be able to generate its replication. Number two, it should be chemically stable and structurally too. Number three, it should provide the scope for slow changes that are required for evolution. Number four, it should be able to express itself in the form of Mendelian characters. If one examines such requirement one by one, because of the rule of base pairing and complementarity, both the nucleic acids have the ability to direct their duplications. The other molecules in the living system, such as proteins, fail to fulfill first criteria itself. The genetic material should be stable enough not to change with different stages of life cycle, age, or with change in physiology of the organism. Stability as one of the properties of genetic materials was very evident in Griffith's transforming principle itself. That heat, which killed the bacteria, at least did not destroy some of the properties of genetic material. This now can easily be explained in light of the DNA that the two strands being complementary if separated by heating come together when appropriate conditions are provided. Further, two complement OH group present at every nucleotide in RNA is a reactive group and makes RNA liable and easily degradable. RNA is also known to be catalytic, hence reactive. Therefore, DNA chemically is less reactive and structurally more stable when compared to DNA. Therefore, among the two nucleic acids, the DNA is a better genetic material. In fact, the presence of thymine at the place of uracil also confers additional stability to DNA. Detailed discussion about this requires understanding of the process of repair in DNA, and you will study these processes in higher classes. Both DNA and RNA are able to mutate. In fact, RNA being unstable mutate at a faster rate. Consequently, viruses having RNA genome and having shorter lifespan mutate and evolve faster. RNA can directly code for the synthesis of proteins, hence can easily express the characters. DNA, however, is dependent on RNA for synthesis of proteins. The protein synthesizing machinery has evolved around the RNA. The above discussion indicate that both RNA and DNA can function as genetic material, but DNA being more stable is preferred for storage of genetic information. For the transmission of genetic information, RNA is better. Five point three, RNA world. From foregoing discussion, an immediate question comes evident. Which is the first genetic material? It shall be discussed in detail in the chapter on chemical evolution, but briefly, we shall highlight some of the facts and points. RNA was the first genetic material. There is now enough evidence to suggest that essential life processes such as metabolism, translation, splicing, etc., evolved around RNA. RNA used to act as a genetic material as well as a catalyst. There are some important biochemical reactions in living systems that are catalyzed by RNA catalysts and not by protein enzymes. But RNA being a catalyst was reactive and hence unstable. Therefore, DNA has evolved from RNA with chemical modifications that make it more stable. DNA being double-stranded and having complementary strand further resists changes by evolving a process of repair. 5.4 Replication. While proposing the double helical structure for DNA, Watson and Crick had immediately proposed a scheme for replication of DNA. To quote the original statement, that is as follows: it has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. Watson and Crick, 1953. The scheme suggested that the two strands would separate and acts as a template for the synthesis of new complementary strands. After the completion of replication, each DNA molecule would have one parental and one newly synthesized strand. This scheme was termed as semi-conservative DNA replication. Figure 5.6. 5.4.1. The experimental proof. It is now proven that DNA replicates semi-conservatively. It was shown first in E. coli and subsequently in higher organisms such as plants and human cells. Matthew Milson and Franklin Stahl performed the following experiments in 1958. They grew E. coli in a medium containing N15 having ammonium chloride, which is the heavy isotope of nitrogen, as the only nitrogen source for many generations. The result was that N15 was incorporated into newly synthesized DNA as well as other nitrogen-containing compounds. This heavy DNA molecule could be distinguished from the normal DNA by centrifugation in a cesium chloride density gradient. Then we transferred the cells into a medium with normal ammonium chloride and took samples at various definite time intervals as the cells multiplied and extracted the DNA that remained as double-stranded helices. The various samples were separated independently as cesium chloride ingredients to measure the densities of DNA, figure 5.7. Can you recall what centrifugal force is and think why a molecule with higher mass or over density would sediment faster? The results are shown in figure 5.7. Thus, the DNA that was extracted from the culture one generation after the transfer of from N15 to N14 medium, that is after 20 minutes, E. coli divides in 20 minutes, had a hybrid or intermediate density. DNA extracted from the culture after another generation, that is after 40 minutes, and it will be second generation, was composed of equal amounts of this hybrid DNA and of light DNA. If E. coli was allowed to grow for 80 minutes, then what would be the proportions of light and hybrid densities DNA molecule? Very similar experiments involving use of radioactive thymidine to detect distribution of newly synthesized DNA in the chromosomes was performed on Vicia faba by Taylor and colleagues in 1958. The experiments proved that the DNA in chromosomes also replicate semi-conservatively. 5.4.2. The machinery and the enzymes. In living cells, such as E. coli, the process of replication requires a set of catalysts. The main enzyme is referred to as DNA-dependent DNA polymerase since it uses a DNA template to catalyze the polymerization of deoxynucleotides. These enzymes are highly efficient enzymes as they have to catalyze polymerization of a large number of nucleotides in a very short time. E. coli that has only 4.6 into 10 to the power 6 base pair. Compare it with human whose diploid content is 6.6 into 10 to the power 9 base pair. Completes the process of replication within 18 minutes. That means the average rate of polymerization has to be approximately 2000 base pairs per second. Not only do these polymerases have to be fast, but they also have to catalyze the reaction with high degree of accuracy. Any mistake during replication would result into mutations. Furthermore, energetically, replication is a very expensive process. Deoxyribonucleoside triphosphates serve dual purposes. In addition to acting as substrates, they provide energy for polymerization reaction. The two terminal phosphates in a deoxynucleoside triphosphates are high energy phosphates, same as in case of ATP. In addition to DNA-dependent DNA polymerases, many additional enzymes are required to complete the process of replication with high degree of accuracy. For long DNA molecules, since the two strands of DNA cannot be separated in its entire length due to very high energy requirement, the replication occurs within a small opening of the DNA helix, referred to as the replication fork. The DNA-dependent DNA polymerases catalyze polymerization only in one direction, that is 5' to 3'. This creates some additional complications at the replicating fork. Consequently, on one strand, the replication is continuous, while on the other, it is discontinuous. The discontinuously synthesized fragments are later joined by the enzyme DNA ligase. Figure 5.8. The DNA polymerases on their own cannot initiate the process of replication. Also, the replication does not initiate randomly at any place in DNA. There is a definite region in E. coli DNA where the replication originates. Such regions are termed as origin of replication. It is because of the requirement of the region of the replication that a piece of DNA, if needed to be propagated during recombinant DNA procedures, requires a vector. The vectors provide the origin of replication. Further, not every detail of replication is understood well. In eukaryotes, the replication of DNA takes place at S phase of the cell cycle. The replication of DNA and cell division cycle should be highly coordinated. A failure in the cell division after DNA replication results in polyploidy. You will learn the detailed nature of origin and the processes occurring at this site in higher classes. 5.5 Transcription. The process of copying genetic information from one strand of the DNA into RNA is termed as transcription. Here also, the principle of complementarity governs the process of transcription, except the adenosine complements now forms base pairs with uracil instead of thymine. However, unlike in the process of replication, which once set in, the total DNA of the organism gets duplicated. In transcription, only a segment of DNA or only one of the strand is copied into RNA. This necessitates defining the boundaries that would demarcate the region and the strand of DNA that would be transcribed. Why both the strands are not copied during transcription has the simple answer. First, if both strands act as a template, they would code for RNA molecule with different sequences. Remember, complementarity does not mean identical. And in turn, if they code for proteins, the sequence of amino acids in the proteins would be different. Hence, one segment of the DNA would be coding for two different proteins, and this would complicate the genetic information transfer machinery. Second, the two RNA molecules, if produced simultaneously, would be complementary to each other and hence would form a double-stranded RNA. This would prevent RNA from being translated into protein, and the exercise of transcription would become a futile one. So this was the part 1 of this chapter. We'll continue the chapter in the next part.